The work of Nobel Prize has transformed our understanding of the world. While approaching the Nobel Prize winners in chemistry, we not only learn their achievements, but also the spirit of honesty, tenacity, innovation, and forging ahead.
Hello everyone, welcome to today's class, Approaching Nobel Prize Winner in Chemistry. 大家好，欢迎走进今天的课堂，走进诺贝尔化学奖。Today I will introduce you Eric Betzig, who invented super-resolved fluorescence microscopy. In 1873, the German scientist Ernst Abbe discovered the diffraction limit of visible light, a well-known formula carved on his tombstone. For a long time, it has been believed that optical microscopy cannot break through a limit because it never achieved a higher resolution than the half wavelength of used light. However, the winner of the Nobel Prize in Chemistry in 2014 skillfully overcame this limit by using fluorescent molecules. Their groundbreaking work has brought optical microscopy to a nanoscale.
I got started in super resolution back in 1982 when I went to Cornell University for graduate school and met my eventual thesis advisors, Mike Isaacson and Aaron Lewis. So Mike had recently developed a means of using electron beams to fabricate holes in a membrane as small as 30 nanometers and then make the membrane opaque. And he and Aaron figured that if we could shine light on one side of the screen, then the light that initially comes out the hole from the other side would create a sub-wavelength light source that you could then scan point by point over the sample and generate a super resolution image. And so the idea was to try to create an optical microscope that could look at living cells with the resolution of an electron microscope. Compared to that, everything else in the department seemed boring to me, so I said, please sign me up because that sounded really exciting. So at that time, many people told us that this idea would never work, either because it violated Abbe's law, or even worse, the uncertainty principle.
For today's class, I will introduce you one of the Nobel winners in chemistry in 2014, who is Eric Betzig. His prize motivation was for the development of super-resolved fluorescence microscopy. He's an American professor and works at Howard Hughes Medical Institute. In a normal microscope, the wavelength of light sets to a limit to the level of the possible detail. However, this limitation can be overcome by methods that make use of fluorescence, a phenomenon in which certain substances become luminous after exposed to light. If you remember, I have introduced you Chen Yongjian's green fluorescent protein in our previous lecture. Around 2000, Eric Betzig and William Monner helped create a method in which fluorescence in individual molecules is stilled by light. An image of very high resolution is achieved by combining images of different activated molecules. This makes it possible to track processes taking place inside living cells. At UC Berkeley, Betzig works on imaging technology, particularly at the intersection of physics, biology, tetrabyte scale imaging analysis. Research in Betzig lab is advancing the understanding and application of the multiple imaging technologies such as lattice light sheet, adaptive optics, electron microscopy, and living cell structure illumination microscopy, and etc. These advances are allowing scientists to review and understand the structure and dynamics of living cells and tissues in detail. The three Nobel Prize winners developed two different and independent approaches for breaking through the resolution limit and achieving super resolution. One approach uses the detection of single molecules and their precise location down to a few nanometers, and a strategy to precisely localize many single molecules and defines a complex structure. The other approach uses pattern illumination to spatially control the emission of excited fluorescence. We first described the single molecule-based approach and continue with the pattern illumination technique. Although the intensity profile of single fluorescent emitter is subject to diffraction, its center can be localized at much high precision. Whereas the size of the emitter molecule is about 1 nanometer, the fluorescent spot produced by diffraction within the microscope is actually about 200 nanometers. Figure 1a and 1b show the intensity profile of such a diffraction-limited signal referred to as the spot spread function, called as PSF, which can be approximated by a two-dimensional Gaussian function. Provided the detected signal is generated by only one emitter, the center of the PSF determines the position of the emitters with very high accuracy. As the number of photons detected from a single emitter increased, and the accuracy improves, and then the peak position of the PSF can be determined. And the position of a single emitter can be localized more precisely. Hence, Abbe formula can be modified to contain an enhanced localization precision, where n is the total number of detected photons in this equation. Thus, for example, within 100 detected photons, the resolution for the localization of a single emitter is increased by a factor of 10. This high-resolution microscopy paved the way for tracking single molecule with nanometer localization accuracy. Whereas 1 emitter is sufficient for single molecule tracking, microscopy of complex structures need many emitters to visualize the features correctly. However, when all the labels are excited at the same time, as shown in Figure 2b, high-precision localization of the individual emitter is no longer possible due to overlapping the PSFs, leading to a blurred or non-resolved image. It was Eric Betzig who first made a valuable suggestion for a solution to this contradiction. His idea to isolate emitters by subspectral property. The separated subsets of emitters each form a sparse set in space. Thus, the position of the emitters with one subset can be determined with very high accuracy. Later on, when Muller investigated the mutant of green fluorescent protein, he encountered a strange photophysical behavior of the T203F mutant. The mutant fluorescent one illuminated with 488 nanometer light and eventually switched into a non-fluorescent dark state. Amazingly, it was reversible photon bleached, but could be reactivated by 405 nanometer illumination as shown in Figure 2c. This finding triggered research for other GFP mutants with similar photon physical switching characteristics. After photon bleaching, new fluorescent proteins are activated in the following flames and cycle starts over again, as shown in Figure 2d here. Finally, one obtained the full complex picture in high resolution by summing up all the highly resolved single emitters of each frame. This significant gain in resolution is best visualized by two images taken from Betzig's landmark publication in 2006, where he introduced the technique with a resolution of about 20 nanometers. In summary, two key elements are required to create a super-resolution image of complex structure by localization microscopy. First, the fluorophore labeling in the structure must be activated controllable in such a way in every image frame only a subset of labels are emitting and their diffraction limit signals are well separated. Second, each fraction limit PSF of a single molecule has to be fitted with an appropriate model function to estimate the position of the emitter with high precision. The collection of molecular positions determined with high accuracy allow then the reconstruction of structures in a point-to-point fashion. So that's today's lesson about super-resolved fluorescence microscopy and its inventors, Eric Betzig. Thank you for watching.
大家好。2016年，白兹格携夫人吉娜一道来中国讲学。吉娜是安徽蚌埠人，毕业于中国科技大学。在加州伯克利大学获得博士学位后，加入白兹格实验室做博士后研究。吉娜是一位物理学家和神经生物学家，成果卓著，两人堪称比翼双飞。白兹格在演讲中坦言，也许是看多了科幻小说，我一直都希望能做出一些东西，看似异想天开，结果却拯救了世界。其他人都觉得是不现实的领域，正是我所喜欢做的。化学认识我，我不认识化学，白兹格说。虽然诺贝尔化学奖委员会给他颁奖，但他实际不懂化学。他是这样自我定义的：我不是物理学家，不是化学家，也不是生物学家，我是工程师，光学工程师。为生物学家开发工具，帮助他们看到活体内的分子。中国科学院物理研究所李明在《超分辨显微至极至美》2014年的诺贝尔化学奖述评一文中评价说，白兹格、赫尔和莫纳将已知的技术推至极限，最早探测到零聚态体系中的单个荧光分子，利用荧光分子的开关效应，加上物理教科书上的受激辐射原理和数据分析中常用的拟合定位方法，绕开了这个似乎不能突破的极限。他们将光学显微技术带入到纳米尺度，引发了常温下活体生物学研究的又一场革命。他们对科学的追求堪称至极至美。在讲学现场，白兹格说出了自己对青年科学家的忠告：不要害怕冒险，不要因为追求安全而搭上别人的便车，要勇敢地开拓属于自己的道路。好了，本次课到此结束，谢谢大家，再见。